So keep in mind, remember how in chapter 1 we talked about things that were prokaryotic and eukaryotic. So everything in that chapter 4, keep in mind, is prokaryotic. Everything in chapter 5 is eukaryotic. And when we get to chapter 6, everything in that chapter is acellular. So that's kind of the scheme of it here. All right, just basically comparing prokaryotes to eukaryotic cells. Keep in mind that we learned some of this already before. Prokaryotic cells lack a nucleus. Eukaryotes have the nucleus. Prokaryotes do not have membrane-bound organelles, whereas eukaryotic cells do. Prokaryotic cells have that peptidoglycan layer, which is the cell wall, the majority of them do. Eukaryotic microorganisms, some of those have a cell wall, and I'm going to let you know which ones those are today. Probably going to ask you a test question about that. But if they do have a cell wall, they do not have that traditional peptidoglycan present. So it's made up of a little different composition. Now, as far as the prokaryotic cell, very small, less than several micrometers. The eukaryotic cell can be 10 times as large. Division. Prokaryotes divide by binary fission. It's a very simplistic process that is similar to mitosis, but there are no centrioles in the cell. You guys remember the centrioles that pull those sister chromatids to opposite sides. Those aren't present. So it is a little more of a simplistic process. Whereas eukaryotic cells, they divide by mitosis, or reproductive cells by meiosis. I don't have to know anything about this slide here. It just shows you an example. This is an animal cell. This picture's not in your book. The green part is the nucleus. Compare the nucleus to the bacterial cell. And you guys remember with the simple saying how small those organisms are. So you just got to adjust to actually looking at such a small organism. Viruses are measured in nanometers. So viruses are not seen with that traditional microscope that we use in the lab. So the optical scopes do not work on it, but the electron scopes you can actually see a virus on. So you got to go to a higher magnification in order to see something with nanometers. So that's why you will never see viruses in this lab, because we can't see them under the scope that we have. Now, this is a basic structure, and I'm going to go through each of these parts individually, so just kind of take a deep breath during this. Prokaryotic cells can have appendages on the outside. So things like flagella, pili. I've got a slide on that, so you don't have to write all this down. They might have something called a capsule. A capsule helps to protect that organism from phagocytosis. It's a type of glycocalyx, which I have another slide on that as well, so you don't have to write real fast during this. The cellular envelope varies between the gram-positive and the gram-negative. And remember that gram-positive, gram-negative have nothing to do with the charge in the dyes. Both gram-positive and gram-negative stain with a positively charged dye. That gram-positive and gram-negative is referring to the differences in the cellular envelope. And that's what we got to learn today. And this will even be on your comprehensive exam, so this is very important that you guys get this structure down. Now, sometimes there's an outer membrane. You'll have a periplasmic space between membranes. You got a cell wall for most organisms under this prokaryotic category. Couple of things like mycoplasmas. There's a few little guys out there that lack a cell wall. And then you have a cytoplasmic membrane. Now in the cytoplasm, you got the nucleoid area. It's where your DNA is just wadded up in there. Ribosomes for protein production and also storage granules. So that's the kind of thing you'd find in the cytoplasm. So the most complex part to this prokaryotic cell is that cellular envelope. So let's start with the outermost layer. If the organism has the ability to produce a capsule, this is called a glycocalyx, or glycocalyces is plural. It can be a capsule or in the form of a slime layer. Either way, the composition is polysaccharide, so make sure you highlight that on your slide. It's polysaccharide. Now you guys remember that negative stain identifies whether an organism has a capsule or not. Those polysaccharide layers are really, really hard to stain. So that's why if an organism has a capsule, you use negative staining, so you stain everything else around it. You can counterstain the organism, but there's a clear area where the capsule would be. So that polysaccharide is poorly, poorly stained. Now, slimy or gummy substance, that's a good description. You guys can see the difference between a slime layer and a capsule. Typically, the slime layer is what you find in a biofilm. So as organisms make communities, a biofilm is just a community of microorganisms, they secrete polysaccharides, and it can vary from organism to organism, but things will stick on, even eukaryotic microorganisms. So for instance, a biofilm you might find in your toilet if you hadn't cleaned it for a while, or around the tub, on your teeth. You guys kind of get the idea. Now the capsule will individually surround one organism. So that slime layer can be secreted and a bunch of things can attach on, and it can encase a lot of different stuff. But the capsule surrounds one thing. Now a lot of organisms that cause, say, pneumonia or meningitis, they produce capsules, and that helps to protect them from that phagocytosis process that we do, so make sure you know that. So protection is good function, but I'm specifically going to ask you, protects against phagocytosis. It also protects against dry mouth, it's for adherence, and it aids in pathogenesis. Now let me talk about that for a second. All of these things that help the organism be pathogenic, we could call virulence factors, makes them virulent. So something that would protect a microorganism from phagocytosis would help it to escape some of our immune defenses, so that would help in pathogenesis. Now, not every organism can produce that glycocalyx. So on the top of that slide, you could put some. Some organisms have it, some are not capable of producing that layer.
Which layer?
The glycocalyx. So if they have it, we got to work a little harder to try to ingest those types of cells. So not everything has that, and that's a good thing. Now, the next layer, if it has a capsule, would be the outer membrane, and that's only in gram-negatives. The gram-negatives have an extra membrane on the outside called an outer membrane, still that phospholipid bilayer. But now there is an endotoxin that we find. This is also considered a virulence factor. That endotoxin is lipopolysaccharide, and it's just part of that outer membrane. There's a toxic portion called lipid A that's just part of the LPS. Now our body specifically recognizes LPS is foreign. We start making immune responses. There's little parts to our immune system that say, okay, that's not on the rest of our cells. That is foreign. Now also in this membrane you'll find porins, nutrients and things have to get in. Lipoproteins will anchor it to the cell wall. And again, like I said, only gram-negatives have this extra membrane. Now the next layer right beneath the outer membrane, if the organisms have that, is the cell wall. The cell wall helps to provide shape. So when I ask you on the exam, what provides shape, that's the cell wall. Helps to withstand that osmotic pressure, so it's protection and shape. Now as far as the composition, it is made up of peptidoglycan, and specifically it's the murine type. There's several types of peptidoglycan. But when you take that word and you divide it up, you can see that that particular peptidoglycan layer is going to be part protein and it's going to be part polysaccharide. And that was the example of a polysaccharide that I wanted you to know for this past test. So it's part protein, part polysaccharide. You have chains of alternating polysaccharides. It's N-acetylglucosamine. I'll abbreviate that NAG. And N-acetyluramic acid, I'll abbreviate as NAM. And then it's cross-linked with the peptides. You guys got this picture in your book. So those, where it says G and M, that's the carbohydrate portion. The yellow linkages, that's the protein portion. Now I will expect you to know that structure for the exam. I love asking a question about the structure of peptidoglycan. Don't know why, I just do. We talk about peptidoglycan in the cell wall an awful lot, so I should ask. Should ask you what we talk about a lot. Now, in gram positives, they have some extra stuff in that cell wall. It's teichoic acid and lipoteichoic acid. So right here, teichoic acid and lipoteichoic acid. They are involved in cell wall maintenance. Now, in gram positives, that cell wall is very thick. Gram negatives, it's very thin. So those gram positives require different components to keep that cell wall in good shape. So all that slide I like for the test, the whole thing. Now let's talk about some of these shapes that the cell wall gives it. Three main shapes. Coccus is singular, cocci is plural. That is a circular or sphere-shaped organism. On the exams, always use cocci. Even on the lab exams, I want you to use that terminology. Now, bacillus or rod, you can use that interchangeably. Now, notice that the bacillus is lowercase, so that's just referring to shape. But now, let's say we have the organism Bacillus anthracis. You guys remember how to write for genus and species. So that B would be capitalized. But from the name, we would assume that it would be rod-shaped. Now, all these other ones go into these spiral or curved forms. Spirilla, spirochetes, vibrios are more comma-shaped than spiral-shaped. Now, where I have antibiotic action, penicillin. Antibiotics, take penicillin for instance, produced by a fungus, penicillin. And it's produced against prokaryotic cells. That's kind of its immune system. That's how it defends itself. So, these antibiotics are targeted towards something that the eukaryotic cell lacks. So we do not have a cell wall. Animal cells don't have a cell wall. There's a couple of eukaryotic microorganisms with a cell wall. But since we do not have a cell wall, this won't affect any of our cells in any adverse way. So makes it a good target. So a lot of things are actually targeted towards destroying that cell wall and preventing it from being synthesized back. Penicillin is one of those. So are the good cephalosporins. There's quite a few things out there. And that's what it targets, that layer. Now, the gram stain, you guys are going to do the gram stain today. Gram stain is a little more complex than the simple stain that you did last week. But the gram stain is the one I'm going to have you do for me on the test. I know you're not going to be a pro at it, but you guys get to play around with it at least. So the gram stain, this one is a differential stain. Two different dyes are used. So in this scenario, the organisms that have a thick cell wall stain purple, and those are called gram positives. The organisms that have a thinner cell wall lose that first color and end up staining with a secondary color, and they stain kind of a pinkish red, so they're gram negative. Now, there are some organisms that have different cell walls, like any of the mycobacteriums, tuberculosis, leprosy. They have that extra component in their cell wall, so it makes them hard to stain with the gram stain. So you can use something different for those guys, which I will talk about that. All right, let's take a look here. See how large that capsule is? That's the yellow part. And that's the part in that negative stain. It'll be clear. Now, the next layer that you get to is the cell wall, cell membrane, all the stuff in the cytoplasm. Would this be a gram positive or a gram negative?
Gram positive is thicker.
It's red.
Well, it's hard to judge. That cell wall's the gram. It's hard to judge. It is gram positive. That's not the answer I'm looking for. It's hard to judge how thick they made that cell wall with that picture. There's one thing that gives it away.
The red? No.
It's missing a layer. Anybody figure out what that is?
It's there. Yeah, I can't see the red.
That outer membrane. So if it were to be a gram negative, it would have an outer membrane right outside the cell wall.
That's got the lipid bilayer, right?
Yep. So they have, the gram negatives have two phospholipid bilayers. You've got an outer membrane and a cell or cytoplasm. We'll talk about the order in just a minute again. All right, taking a look at these three main shapes. When you look at the cocci, you might see them by themselves. You might see them as a pair, which is called Diplococcus. Tetrid, Streptococcus is in chains, Staphylococcus is in clusters. Now these are arrangements, so how they actually divide is how they're going to look under the microscope. So pay close attention to Streptococcus, you're always going to see in the chains. Staphylococcus, you're always going to see in clusters, looks like grapes. But make sure you know the different arrangements that these organisms can have. Some pictures, and you can definitely see the chains versus the clusters. And not always are the clusters in big clusters. It's one of those things. Now the rod shaped again, you can see them by themselves, in a pair, or a chain, Streptobacillus. Spiral shapes, all of those wonderful spiral shapes fall under. Now your book groups its spiral and curved forms. The curved is for the Vibrio. There are some other crazy shapes that you'll find. I'm not going to ask you that on the test, especially with archaea. Archaea are another type of prokaryotic cell typically studied in an environmental microclass. Find them in very extreme environments, but they are prokaryotic and they have some odd shapes to them. Typically, they're not pathogenic, so they really don't fit into this type of class as well, but just to let you know. Now, your textbook has changed this a little bit. It's okay that you have a different picture, but know those three main categories: cocci, rod or bacillus, curved or spiral forms. They made it just a little bit different in your book, so it's okay. Now, one of the things that's interesting is this group called Coccobacilli still fall under the rod. They're almost like ovals, but they still have a little bit of an elongation, so that's why they sit there. All right, the last layer in the cellular envelope. The cytoplasmic membrane, sometimes called the plasma membrane, sometimes called the cell membrane. You guys have probably used all those different terms. That contains the cytoplasm. Both prokaryotic and eukaryotic microorganisms have this layer. Remember, it's selectively permeable, something that you guys learned back in A&P. So it regulates what goes in and out of the cell. That phospholipid bilayer, part of it being hydrophobic and hydrophilic, helps to regulate that. Now there are proteins associated with the membrane so that things can get through that are hydrophilic, for instance. There's also receptors and so forth. Now, fluidity of the membrane. Remember, the membrane is very flowing, kind of like a waterbed. Cholesterol does help to stabilize that membrane some, but it is fluid. Now, we need to stop and summarize these layers here. So the gram-positive versus the gram-negative. Now, I always say the gram-positive looks like that open-faced sandwich, which I've never heard of until I came to Ohio. I'm not really a big sandwich person, but I get it. Was it turkey shots or something? They're pretty good. I just don't like white bread. Gram negatives, that's more like the traditional sandwich. So, from out to in, the first layer in the gram positive, and this is the cell wall, that thick layer of peptidoglycan. The first layer is peptidoglycan. Working your way in is the cell membrane. So gram positives, outermost layer cell wall, cell membrane, innermost layer. Gram negatives, outermost layer, the outer membrane. This layer right here is the cell wall where it says peptidoglycan. And then the most inward layer is the cell membrane. So that cell wall is sandwiched between two phospholipid bilayers. So everybody got that. It's important to know the layers. So from out to in, gram positive is cell wall, cell membrane, right? Gram negative out to in is outer membrane, cell wall, cell membrane. Now, what if it had a capsule? Where would that go?
Then you're in the whole thing.
Right, on the outside of everything. But remember, that may be present and may not be. But if it were there, yes, it'd go on the outside of everything. Right. Okay. Now, some of the appendages that we find on the outside. Bacteria can have pili. And I'm also going to talk about fimbria. Wait till I get to that slide. Some textbooks used to put pili and fimbria into the same category. This book distinguishes between, so we're going to distinguish between the two. And then flagella. Now, there isn't really a rule. They can have one, they can have both, or they can have none. So again, it's a maybe-maybe. Now, distinguishing between pili and fimbria. Now, the purpose of both is for attachment. But go over to that where it says purpose of fimbria. Main purpose there is to attach to the host. So I've given an example here, adhesion to the host epithelium. Used for cell-to-cell contact. And a lot of times there's recognition sites that they'll know to attach to.
So on this side, it's
That's
for
pili.
both of them?
Now this side will be for
Just the
pili.
pili?
Yeah, so put pili at the top there.
Okay.
Both are used for attachment, and that's why I didn't stick it up there yet. But I've got attachment over here in this definition. So now when you get to gram negatives are the only ones at this point that they found to mate or conjugate. And it's not like you think. Bacterial cells typically have circular chromosomes, but they might have something called a plasmid. A plasmid is extra chromosomal information that encodes for some sort of resistance factor, like resistance to antibiotics. So what happens is the organism replicates and then sends that information through something called the sex pills. A lot of times they'll call it the F. F pillis or sex pillis. I like to use the term sex pillis. So that is for the transfer of DNA. That's their conjugation. It's for some sort of resistance factor. It's not because they're going to mate and have babies. They just passed on their DNA. Gram negatives at this point are the only ones that can do that. I'm sure that something will occur at some other point in time where we find that other organisms actually do this as well.
Oh, Lord.
And this picture at the bottom, I can't remember if your textbook still has this. I think this was from the first edition. You can see the fibria along the outer portion of that cell. They're a little shorter, and the pili are actually attaching to other cell types that are bacterial cells. And the same thing goes up here. This is the fimbria that they're trying to show you there. And then they've got the sex pilus connecting to the other bacterial cell. Oh yeah. That was an old picture. They called that pilus instead of they used to not distinguish between those two, but now they. Alright, the next appendage, flagella. Now keep in mind that this is for motility. So not all bacteria are motile. Some are motile, some are non-motile. So again, this might be something that they have, might be something that they don't have. Now this is also considered a virulence factor because it can move that organism from a less favorable environment to a more favorable environment. So for instance, things that can cause UTIs, a lot of those organisms that are transferred from one area where they're normal flora to an area where they're not have flagella. They just move on up. Now, a lot of times they'll use the term chemotaxis, and that term is meaning that they can chemically taste their environment, and if it's non-nutrient rich, they'll move somewhere else. So taxis, like it's moving, like taxing. Now look at the arrangement on the flagella. We'll learn this again in Lab 3. But these organisms have different arrangements. Sometimes they only have one flagella, and that is monotrichous. Sometimes they have flagella all the way around the cell. That's called peritrichous. Sometimes they have like a little tuft on the end, kind of like you pulled your hair back in a ponytail. It's like a ponytail flagella. That's called lophotrichous. Now there's another one in lab called amphitrichous that I'll show you with one flagellum on each end. So there's some different arrangements. I want you to know the terminology as far as that goes. Polar and non-polar flagella, that's just whether it's evenly distributed or not. I won't ask you that whatsoever. Now the three main parts I do want you to know, that is the basal body, and you can see that that's wedged down in the cellular envelope, the hook, and the filament. Now as far as the movement goes, I don't really get into a lot of this, but it can vary between microorganisms. Now, the three terms, you don't have to know magnetotaxis, but know the top three. As far as movement goes, that chemotaxis, that means that the organism is sensing its environment, whether it be chemicals or nutrients. And if it has flagella, it can move towards a more favorable environment. Phototaxis, it will sense light intensity, so depending upon what the organism needs, it can again move to a favorable environment. Aerotaxis, different organisms need different amounts of atmospheric oxygen. Some cannot use it, some can, some need small amounts. Now with that being said, flagella also allow it to move towards a more favorable oxygen concentration, and that's called aerotaxis. So know those three terms. I'll pick out one of them for these. Now as far as the cytoplasm, you pretty much got a lot of water in there. Site of metabolism. The nucleoid is the region that you'll find the DNA in. Ribosomes are the site of protein synthesis. And then you have some inclusion bodies. I'm not going to ask you about those, but the Other ones I most likely will. Now the ribosome, I tend to focus on a little bit more. The ribosome, together the whole thing measures 70 Svedbergs.
70 what?
You don't have to know the Svedberg, it's just 70S. It's kind of how it works. A little different method of evaluation than traditionally. Now, this ribosome can break apart into a large and smaller subunit. They have little different numbers. 50S is the larger subunit. 30S is the smaller subunit. I want you to know the measurements for those. Now, for the eukaryotic cell, the overall measurement is 80S. Now, I'll probably ask you this on the test.
What was the smaller unit? I'm sorry.
The smaller unit for the prokaryote is 30.
30.
So the eukaryotic is 80. The prokaryotic large is 60. Or the eukaryotic large is 60.
So,
I'm getting distracted by
I
everything.
know, he's just.
All right.
Yeah.
That's why I said he thinks
All right,
he's in charge.
eukaryotic. Prokaryotic numbers are 70, 50, 30. Eukaryotic numbers 80, 60, 40. I couldn't remember if I just said pro or eu, but when I changed it, repeated myself. So I had to clarify. So prokaryote 70, 50, 30. Eukaryote 80, 60, 40.
Just a tad.
Now this is unique because that prokaryotic cell has a different ribosome size than the eukaryotic cell, meaning that antibiotics can target protein synthesis without affecting protein production in a eukaryotic cell. So a lot of those antibiotics that we take, like tetracycline for instance, interferes with protein synthesis.
Say that again, would you? The whole perfect. Yeah.
Right, so some of those antibiotics target protein synthesis due to the fact that the ribosome size is different. So you can interfere with a prokaryotic cell without doing anything to eukaryotic protein production. So tetracycline, it's a good one. Chloramphenicol, there's quite a few in that category that target protein synthesis. Okay, thank you. All right, remember me talking about endospore formers, and endospores are very hard to kill. There are some types of bacteria that have the ability to produce endospores. So one of the ways you can think of it is they're like hibernation structures. So if they are exposed to some type of unfavorable condition like extreme heat, for instance, that endospore will form. Now they can survive in that form for hundreds of years. So it's just like a protection. It's almost like they, reminds me of some sort of movie where they put like armor around themselves or something. Like, what was that, Austin Powers, where the mean dude always went out into space. Anyway, these steps in sporulation, your book is very descriptive and detailed. I'm not going to ask you step number 1, step number 2. I just want you to know basically what's happening during this process. So if you take a look at that top cell, you have unequal cell division. Now, you've had to replicate this DNA, so that DNA is in both parts. You need an exact copy because eventually that endospore should function like that cell did before. So you're just preserving it. Now, as it divides, you'll have a vegetative portion and something called a forespore. Sometimes textbooks call it a prospore. That will eventually be the endospore. Now, there is a very thick wall that forms, and it is peptidoglycan, but it's a little different than what we had talked about. It's more keratin-like. So it'll cover all of those parts of the spore. That vegetative cell will lyse and release the endospore. And if those conditions are harsh, the vegetative cell dies off. And then this is going to just hibernate until things become more favorable. We'll get there. So you still got to replicate that DNA and pass it on now.
I'm struggling.
I feel like I'm competing. Should it, you want me to stand over here so I can kind of?
Maybe that would
I get
help.
distracted just a little bit. Yeah, maybe I need to talk this way. I know, I'm getting a little distracted. Anyway. So you got to have DNA in both parts. You want that spore to eventually germinate and act just like that regular cell did. And this is those general steps there that I'm not going to ask you step 1, 2, 3, 4, 5, 6, 7, 8, 9. Now this you do not have to know. I just want you to understand why things look the way they do under the scope. Bacterial cells divide by binary fission. I haven't gotten into that process yet. Like I said, it's very simplistic compared to mitosis. But how they divide is how they're going to look under the scope. So Streptococcus actually divides along the same plane, and that's why it's in chains. You go down to Staphylococcus, and you see these 3 different planes. It divides along irregularly. That's why it ends up as clusters. So it's just the way it divides is why it looks the way it does. Eukaryotic cells. I'm almost done with this chapter. This stuff's easy stuff here. We went through this in AMP1, and we went through Torrey with Meisner. All the parts are still parts. Now I gotta make it a little micro because we got some eukaryotic microorganisms. So there's some things I do want to point out. As far as some of the appendages, eukaryotic microorganisms can have flagella. Of course, it's for motility. It's a little more of a wave-like motion than propeller-like on a lot of prokaryotes. As far as the composition, it contains microtubules. That's as far as you want to go. Now also they might have cilia. With eukaryotic microorganisms, it's very similar to flagella. They are shorter and they are more numerous than the flagella. Now also a couple of microorganisms that are eukaryotic. There are quite a few fungi that have glycocalyces. But they also might have a capsule. So that glycocalyx, that's a capsule. Again, it's made up of polysaccharides. The functions, protection, adherence. And also with these eukaryotes, it recepts signals from other cells. But now it's still going to do the same thing, protect that fungus from phagocytosis once it's in the body. Now, most eukaryotic cells don't have a cell wall that we've talked about in the past. Some of these microorganisms that we'll talk about do have a cell wall. Now, if they have a cell wall, there's a lot of diversity as far as the composition. I do want you to know the chitin mixed glycan, so highlight that part. Instead of it being peptidoglycan, this is what you typically find in a eukaryotic cell wall. Yeah, right here. So typically that cell wall you don't find, but if you do, it's got a little different composition, not peptidoglycan like you think. Now, animal cells do not have a cell wall. We don't have a cell wall, which makes, like I said, all those cell wall inhibitors really good for our particular cells. Now, the type of eukaryotic microorganisms that might have cell walls, fungi and algae. So you know those two. I will most likely ask you that on the test. Fungi and algae. So now, even a fungus that produces penicillin, that's still against that peptidoglycan layer. So since that cell wall is so different, their cell wall is so different, it's not going to do anything wrong. So it's specifically targeted towards that prokaryotic peptidoglycan. Now, as far as the cytoplasmic membrane, that's the same cytoplasmic membrane that you guys learned in A&P. So still that phospholipid bilayer with proteins associated. All right. So from the outside in, you might have a couple that have a glycocalyx, such as a capsule. You have fungi and algae that have cell walls, but the majority of the eukaryotic organisms do not have a cell wall. All right. Now, the cytoskeleton, they call this the framework of the cytoplasm. For a lot of these eukaryotic organisms, it helps in movement so that all those parts of the cell move in somewhat of an orderly way. Now, the term cytoplasmic streaming, that's talking about moving and nutrient distribution. Now, the three protein structures that make up this framework are microtubules, microfilaments, and intermediate filaments. I'm not going to ask you all the other stuff about them. I think you guys covered that in A&P pretty good. So, I'm good there. And if you take a look at it, it does look a little bit like a highway, almost, like a network there. So again, just to reinforce here, cytoplasmic streaming helps nutrients move throughout the cell and also allows some of these eukaryotic organisms to move. Right, the nucleus. As far as we know, it contains DNA. We're good there. There is a nuclear membrane, which is a lipid bilayer. There are pores associated with that membrane. Remember that messenger RNA is going to have to get out of there so that it can bind a ribosome and the protein can be made. The nucleoplasm, that's just the matrix. The nucleoli, remember that real dark spot you find in the nucleus. Masses of RNA and protein. Same old that it was in A&P. Cytomembrane system. You guys remember there was rough ER and smooth ER. It's for sorting and transportation of molecules. The rough has ribosomes associated, the smooth no ribosomes, and specifically the smooth ER specialized in lipid synthesis. The Golgi apparatus, always described as stacks of flattened membranous sacs. That's where molecules are packaged and modified. And I like how they show these vesicles here, those little bubbles that pinch off. They're used for transport. So it could be from things going from the ER to the Golgi or wherever they're to be sent to. Could be inside of the cell, it could be outside of the cell. So things might have to fuse with that membrane or leave by exocytosis. So vesicles, just transport. Mitochondria, chloroplast, you guys know this for energy production. Chloroplast specifically for photosynthesis, you find those in algae and plant cells. And I really won't get into photosynthesis this term, but just to give it a little bit of credit. When we talk about these structures, mitochondria have an outer and inner membrane. And a lot of times they're compared to a gram-negative bacteria. And scientists have actually studied this and came up with a couple of different genus and species that the mitochondria might have been descendants of. So theoretically how that happened was a prokaryotic cell lived with a eukaryotic cell. And then it became its energy producer. So as far as what resembles a prokaryotic cell in a eukaryote, that'd be the mitochondria. Now the mitochondria, that's the place for cellular respiration, contains its own DNA, and it divides independently of the cell. So it's got a little bit of self-sufficiency there. We don't have to go through diffusion and osmosis and all this stuff. You guys okay with the terms endocytosis, exocytosis? I don't wanna ask these on the test because they've been asked and asked and asked and asked. Everybody comfortable with the difference between phagocytosis and pinocytosis? One's just taking in debris or something solid, whereas the other one, pinocytosis, is taking in fluid. Mitosis, meiosis, we're good. You guys don't have to worry about those. We've learned that quite a bit in NLP. You guys want about